Hello and a warm very welcome everyone to our today's webinar on bioanalytical aspect of BA studies for bioanalytical for NC molecule and case studies. The case studies will be covered in upcoming webinar. I am Hemal Kurani, Senior Group Leader, and I represent Veeda Clinical Research Limited, where my role is to serve as scientific as well as technical lead for biopharmaceutical companies. I have over 16 years of professional research experience of regulated bioanalysis in developing, validating, as well as applying bioanalytical method to support clinical studies of generic products as well as new chemical entities. Veeda Clinical Research Limited, together with its subsidiary Bioanalytics India Private Limited Bioanalytics and its joint venture Ingenuity Bioscience Private Limited, together referred to as Veeda Group, offers a comprehensive portfolio of clinical, preclinical, bioanalytical service to support innovator, biosimilar, as well as generic drug development program for our global clientele. Together, we serve clients globally in the following industries like pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbals, nutraceuticals, and as well as medical devices. So, as your facilitator, today I would like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by the panel discussion. We all running a live question answer during the webinar, which will be led by our backend team present with us here. So for all your questions and queries, we have enabled our ask a question feature. It's right on top of your screen, so you can drop your questions in there. Now, I would like to introduce you to our presenter for today's sessions, Mr. Jairaj Chudasma. Mr. Jairaj Chudasma is working as a senior group leader at Veeda Clinical Research Private Limited, where his role to serve as scientific technical lead for biopharmaceutical companies. Jairaj has over 17 plus years professional research experience of regulated bioanalysis and developing, validating, and applying bioanalytical method to support clinical studies of generic products and new chemical entities. He plays a significant role in the developing, validation, and applications of bioanalytical assay to comply with scientific and regulatory requirements. He is having exposure to develop and validate more than 20 NC molecules and more than 200 generic molecules. These methods were successfully applied in clinical studies for regulatory submission. Jairaj has published several research articles in peer-reviewed scientific international journals. So finally, we are beginning with webinar. Over to you, Jairaj.
Thank you, Amal, for the nice introduction. Welcome you all in today's webinar for bioanalysis of NCE molecules. So we all very well about the handling of generic molecules, but the handling of NC molecules are a bit different, and there are certain challenges involved that I will discuss during this session. So here is the outline of today's presentation. NC development process followed by the clinical development stages, role of bioanalysis in pharmacokinetic study in different stages of clinical trials, study initiation workflow from sponsor perspective, NC bioanalysis that include the method development, method validation, study sample analysis and its challenges, and finally the Veeda's experience and expertise in NC molecule. NC development process. The drug discovery and development is the process of generating compounds and evaluating all their properties to determine the feasibility of selecting one novel chemical entity to become a safe and efficacious drug. There are 5 critical steps in the drug development process, including many phases and stages within each of them. The different steps are discovery and development, preclinical research, clinical development stages, regulatory review, and regulatory post-market safety monitoring. So next slide, I start with this clinical development stages. In this stage, the human testing of experiment drug is began in the 4 different phases. Each phase is considered a separate trial, and after completion of a phase, investigators are required to submit their data for approval from the regulatory before continuing to the next phase. So during the entire phase of clinical trial, the bioanalytical techniques are required continuously throughout the preclinical and clinical drug development phases into post-marketing support and may sometimes extend into clinical therapeutic drug monitoring. So the different 4 phases, clinical phases, in phase 1, the safe dose is established for the drug that may be on around 20 to 80 volunteers, while in phase 2, here it demonstrates the efficacy and side effect of the drug on around 200 to 800 patients. In phase 3, safety and efficacy is monitored in large population of patients that may be around 1000 to 5000 patients. And phase 4 is for post-marketing surveillance trials. Role of bioanalysis in pharmacokinetic study in different stages of clinical trials. So why bioanalysis is important at various stages of clinical phases? Here we have to quantify our drugs in biological metrics. So at different stages, we have to extract the information, performing the bioanalysis, and the data obtained during the initial trial can further applied for the design and interpretation of the next study phase during the drug development process. As the data generated during this stage are much important, so PK sample analysis required careful planning with logistics between the clinical site, sample shipment, sample analysis, and PK calculation. So for such careful planning and considering the requirement of PK results, we have to establish suitable SO methodology, which can produce accurate and reliable result for the PK samples of clinical trials. So next is study initiation workflow. This slide is for sponsor perspective to decide the workflow from CRO selection to start the method development of NC molecules. So first CRO selection by sponsor, then the qualification of CRO is planned and they evaluate whether the CRO is able to perform the trial as per the quality standard or not. After qualification, sponsor has sent the request for feasibility assessment. During this stage, sponsor may share some limited information for the molecules, and based on that information, feasibility assessment is done by the CR. Once the feasibility assessment is completed, the confidentiality agreement is signed off between the sponsor and CRO. Then reference standard input license application to get the standard at CRO site. After having this reference standard, the NC method development can be initiated. Bioanalysis of NC molecules. This includes the method development of NC molecules, method validation of NC molecules. Method validation may sometimes extend to cross validation and partial validation as per the requirement. Then analysis of study sample by validated method and incurred sample reanalysis. So next slide, I will discuss So one by one, this method development, method validation, and study sample analysis and the challenges. Bioanalytical method development of NC molecules. So for the NC molecules, the rugged and reliable method is required because sometimes after validation, few changes are required in validated method for some additional testing like drug-drug interaction or metabolite estimation. So the method should support such changes. So we do not have to perform the entire method development activity all over again. Some critical steps of method development initiation, that NC molecules physicochemical properties, it is not available in many cases. Literature on pharmacokinetic based on the proposed dose. If available, we can refer the some stability data of molecules. Then some known metabolites information are available. It will be helpful to design the method development trials like glucuronide and N-oxide. Then the selection of calibration range. After calibration range, we can have to select this internal standard, mobile phase and column selection, and sample extraction procedure. Now, what are the challenges to develop the NC molecules? There are several challenges involved. See, like in generic molecules, we have almost some basic information with us when we start the method development activity, like compound physicochemical properties, solubilities, molecular structure. So it is easy to establish some initial method development trial. But in case of NC molecules, there are very limited information are available. So it has some challenges to decide initial method development workflow. Expected metabolite should be tested during development trial if some information are available in advance. And rugged and reproducible method is necessity for any NC program. The major challenges, that is selection of dynamic linearity range. So in most of the clinical trial, we have to set the calibration range in such a way that it covers minimum dose level to multiple ascending dose levels. So this requires dynamic linearity range. So this dynamic linearity range itself have challenge because of the instrument limitations. The work with such dynamic range, like more than 2000 times, is a big challenge. So in some cases, after establishing the such dynamic range, during clinical study sample analysis, some concentrations still found beyond the validated ULOQ sample. So sample dilution is required. It generally for 100 or 1000 folds. So dilution with such higher amount of sample itself have challenge sample volume for analysis. So here, when the clinical trial is planned on specific study population like pediatric patients, so we can draw a limited blood. So we cannot keep the sample volume above 50 microliter. In some case, we have to set the assay with less sample volume, maybe 10 microliter, 20 microliter. So to work with such low sample volume, it requires sensitive machines and its appropriate handling during the analysis of study samples. In such case, sometimes new techniques such as capillary, dried blood spot cards or tap device also recommended to use for specific studies. Metabolite determination is challenged, method development for active metabolite, some exploratory estimation of metabolite in study samples, and the determination of concentration and correlation with the metabolism. Method reproducibility. Method reproducibility in terms of lab variation. See, the clinical trial duration are almost about 3 to 4 years. So we have to develop a rugged method that it can easily work in multiple labs. If required, the multiple analysis can be planned in multiple labs. So such variation can be minimized if we have developed a rugged method. Sometimes there is a need to involve multiple labs in some trials. So cross-validation is mandatory to present the data reliability between the labs. Then long-term metric stability. As I mentioned that the entire duration of clinical trial is 3 to 5 years. So we have to establish the LT stability to cover the entire study duration. So sometimes if stability is an issue, then we have to add some stabilizer. But here, considering some limitation at clinical side, we have to select the stabilizer very carefully that it can be easily available at site. Method activeness, frequency. So method should be active and frequently tested for its performance because during the clinical trials, the sample analysis is planned on regular interval. So method should be tested prior to estimation of drug during different clinical phase. Maybe we can perform one P&A batch prior to analysis to avoid some repeats. So method activeness, we have to check it frequently. Method validation of NCE molecules. The developed method is to be validated as per the current regulatory requirement of US FDA, EMEA, and Visa. During method validation, we have to evaluate all these parameters like selectivity and sensitivity, precision and accuracy, matrix effect, drug-drug interaction, concomitant medication experiment. If we have some prior information of concomitant drug, that experiment can also plan during method validation. Stability studies, batch size experiment, dilution integrity and recovery. Cross-validation as per the requirement. As I mentioned earlier, then where multiple labs are involved for the analysis of study sample, then cross-validation between the labs are required. Apart from this parameter, some additional parameter also needs to be checked as per the requirement, like stability as specific storage condition, maybe at refrigerated, whole blood stability. Sometimes at clinical site, some refrigerated centrifuges are not available. So we have to establish whole stability to mimic that conditions. Some drug-drug interaction studies. So these are the some additional experiments that can also be performed during method validations. And if any sponsor specific requirement is there, then that also we need to take care during the method validations. So all the method validation experiment we have to carefully design for, and its acceptance criteria we have to clearly mention in the protocol. Okay, after successful completion of method development and method validation, this method can use for the study sample analysis. So in this slide, the study's flow is presented, means study samples flow when the samples are received from site to CRO and then sample received to final report sign off. So once the samples are received, the sample management team stored the sample at specific storage condition. Then the study design is created in Watson LIMS software. At Veeda, we are using LIMS software. Then updation of study sample in the Watson LIMS software for the analysis. Next, we have to prepare batch sequence and work list for the analysis of samples. This batch sequence and work list are further uploaded in LCMS software. for the sample preparation and analysis. These results are imported from LCMS system through digital interface or common share folder. Then results are scrutinized through Watson LIMS for its acceptance, data compilation and verification by group leader. After that, report preparation, the report review, QC review, QA review, and draft report shared with the sponsor. Any comments from sponsor that has to be taken care. And after resolving all this comment, final report is sign off. And finally, the archival of raw data of LCMS, LIMS data, and sample receipt data. So this is the general flow of study sample. Like method development, here for the analysis of study sample, there are certain challenges, logistics and timeline challenges, like sample management, reference standard management, sample result timeline, fast switching and decision making, communication and data flow. So sample management during transit phase, when the samples are shipped from site to at CRO, coordination between the sites and sample management team and custom sample management team and courier agency is required. We have to submit certain document to import the samples at lab. Certain declaration for safety and sample integrity is required. Any inappropriate or incomplete document, there may have a chance that sample can stuck at custom clearance, which overall delay the trials timeline. Reference standard management. The application of import license and approval process against the custom clearance documentation, appropriate documentation is required to get the reference standard. Its accountability of reference standard here, in many cases, the reference standard have very short expiry and limited quantity. So we have to use this standard very carefully, and due to its short expiry and its continuous usage throughout the clinical trial, one has to communicate it with sponsor in timely manner to get the retest COI, so we can meet the sponsor timeline for the analysis of clinical study samples. Sample results and timeline. For first in human study, the timeline are critical to share PK data along with safety profile review for further decision on trials. So in like as I mentioned that in first in human study, the data obtained during initial trials that are much important because this data are required to interpret and further proceed for the next phase. So in this case, the turnaround time from sample receipt to analysis is 3 to 4 days for around 200 samples. If more samples are then we have to involve the multiple machines to meet the timeline. Fast switching and decision making. In some cases, sometimes the predicted range does not work. During analysis, it shows that this calibration range is not working. Many samples concentration found above the limit or below the limit of quantification. So real faster switching is required. Immediate decision is required whether it needs to update the method or we have to redevelop the entire method. Communication is the key parameter. Effective communication during the entire clinical phase trial is much required. Faster communication is key for any successful trial. So here PK scientists should be prompt to revert the trial leads for required information. Any miscommunication leads to the wrong interpretation and delay in trial related decision. See, in some safety point of view, when we analyze the sample at particular phase of a clinical trial and some abnormal results are observed, then CRO must be communicated promptly with sponsors so they can further decide the drug dose and accordingly the next phase of clinical trial is decided. Data flow. The generated PK data should be reliable, acceptable as per the global quality standard. The longer study duration and reliability is the key concern for any follow-up trial decision. Any misinterpretation should be communicated in appropriate forum to further avoid any wrong trial decision. So these are the some logistic and timeline challenging when we handle the study sample for NC molecules. Now the execution of study sample analysis for NC molecules. Prior to start the study sample analysis, clinical study protocol and other documents thorough understanding and training is required for study personals. Procurement of reference and working standard. In case of required, we have to take import license permit to get the reference standard in timely manner. If in some case non-availability of label isotope compound, we have to select the internal standard, and it is also required to get the COA with all required informations. So all this information we have to have prior to start the study sample analysis. Understanding of sponsor specific requirement. So before starting the clinical study sample analysis, if any sponsor specific requirement is there, then we have to take care of particular requirement and same needs to be mentioned in the bioanalytical study plan. Immediate slotting and prompt communications. As the data generated during the different clinical phase are important, so sometimes the samples are received at CRO, it required immediate slotting, and the results obtained during this trial we have to promptly communicate it with the sponsor. To execution of the overall study sample analysis, dedicated and trained sample management teams, LIMS operator teams and report writer team is required, who can understand the sponsor requirement and share the data as per the sponsor specific requirement. Again, sample handling by the trained analyst and the custodian team. Here the sample identification and verification is much important. During receipt of samples, any discrepancy is observed, then sample management team has to communicate with the site. Any sample discrepancy should be promptly communicated with the sponsor. Coordination between operation teams and LIMS operator is required. During analysis, if any abnormal observation, then operational team has to intimate it LIMS operator, who can further communicate with the sponsor. Some identification issue like that. During analysis, based on the initial result, if higher concentration are observed, then pre-dilute the samples. Result sharing through secure digital interface to maintain the confidentiality. After sample analysis, report preparation and report review as per the sponsor specific template with eCTD, and finally we have to archive the raw data and electronic data. Here is the Veeda's experience and expertise in handling of NC molecules. At Veeda, our bioanalytical lab have a vast experience for the handling of different NC molecules. All the scientists have science and pharma postgraduate, and senior person have more than 15 years of experience in bioanalytical field. We have developed and validated around 8 NCE molecules in different biological metrics like plasma, blood, urine, skin, colon tissue, and contact lens. We provide the service in wide variety of metrics like plasma, whole blood, urine, skin, and colon tissue. Veeda has analyzed 200 samples and coordinate with more than 200 sites across the globe. The team is well trained and understand the requirement of sponsor. So data transfer as per sponsor specific requirement. Veeda's familiarity and compliance with the regulatory requirement of US FDA and other agencies enable our clients to have a complete confidence in the quality and acceptability of analytical data. At Veeda, we have a latest instrument. We have all the range of equipment from low end to high end. We have a total 49 LCMS machines, so we can slot the study sample analysis immediately and provide the result promptly. At Veeda, we have a Watson-Leach software, that is laboratory information management systems, which prevent the human error, and it significantly reduce the result sharing timeline. So here I am ending this session. Next we have a panel discussion where we have a question and answer. My colleague Mr. Hemal Kurani will take us for panel discussion. Over to you, Hemal.
Thank you, Jairaj, for nicely covering various topics like NCE molecule execution workflow, challenges for method development and clinical study sample analysis, depth explanations regarding bioanalytical workflow during method validation, as well as study sample analysis of NCE molecules. Now, moving on, we will be proceeding ahead towards our panel discussions round, where we are going to answer some of really interesting questions posed by our audience. My first question is for Ms. Swati Gutikar, but before asking questions, I would like to give a little introductions of her. Ms. Swati Gutikar is Head of Bioanalytical at Veeda Clinical Research Private Limited, Ahmedabad, India. She has more than 30 plus years professional experience in bioanalytical research. Her role is to guide bioanalytical scientists for method development as well as method validation activities and supervise activities performed in regulated bioanalysis. She has published several research articles in peer-reviewed scientific international journals like Bioanalysis, Journal of Chromatography-B, Journal of Chromatographic Science, Journal of Pharmaceutical Analysis, as well as Biomedical Chromatography, etc. So moving to my first questions, the first question is to Mr. Swati Gutikar, and the question is that how to start method development activity for NC molecules where the molecule informations are quite limited. Over to you, Ms. Swati Gutikar.
Thank you, Hemal, for the nice introduction. Thank you, Jairaj, for the wonderful presentation. As you have asked the question on the development part of the NC molecule, as you all know that the development of NC molecule is quite challenging because very less information is available of the molecule, like the molecular structure or its physicochemical properties. So we have to rely on the very limited information provided by the sponsors. So if we have the molecular structure available with us, then we can select the column and the proper mobile phase for it. And based on the initial results and its interpretation, then we can have the development strategies moving forward. So if we have the solubility also, then it plays a key role in having selecting our extraction methodology. And the preclinical data which the sponsor provides to us is useful in selecting the calibration range for the predefined dose. So whenever we have to develop a method development for the NCE molecule, we have to take the trials, and based on its results and its interpretation of data, we have to move forward with the development part. Thank you.
Thank you, Ms. Swati Gutikar, for answering the questions to initiation of method development activity of NCE molecules. Now, my second question is for Dr. Ajay Gupta. So before asking questions, I would like to give a little introduction of him. Dr. Ajay Gupta is working as bioanalytical investigator at Veeda Clinical Research Private Limited. He has more than 20 plus years of professional experience in bioanalytical research. His role is to guide bioanalytical scientists for method development and method validation activities and supervise activity performed in regulated bioanalysis. He has published several research articles in peer-reviewed scientific international journals like Journal of Chromatography-B, Journal of Chromatography Science, as well as Journal of Pharmaceutical Analysis, Drug Testing and Analysis. So my second question to Mr. Ajay Gupta is that how to set turnaround time for the first in human study? Over to you, Mr. Ajay Gupta.
Thank you, Hemal, for giving me an opportunity to answer these questions. But the timeline is the most critical part for the molecule tested first in human because the initial data is further used to design an interpretation of the next phase of the study. Effective communications between the sample management team and site is required for the immediate slotting and result sharing. Robust and reproducible method is required to avoid failure experiment because failure of experiment may impact overall study timeline significantly. So if we are talking about the timelines, in most of the cases, the turnaround time is 4 to 5 days from sample receipt to analysis, which required careful planning and trained manpower to execute the analysis. Thank you, Hemal.
Thank you, Mr. Ajay Gupta, for swift answering the questions. Moving to the third questions for Dr. Vikas Trivedi. And just moving to the third questions, before asking questions, I would like to give a little introductions of him. Dr. Vikas Trivedi is working as bioanalytical investigator at Veeda Clinical Research Private Limited, Ahmedabad, India, where his role is to serve as scientific and technical lead for biopharmaceutical companies. Vikas Trivedi has over 17 plus year professional research experience of regulated bioanalysis in developing, validating, and applying bioanalytical method to support clinical studies of generic product as well as new chemical entities. He plays a significant role in the development, validation, and applicable of biological assay to comply with scientific as well as regulatory requirements. Dr. Vikas Trivedi has published several research articles in peer-reviewed scientific international journals like Bioanalysis, Journal of Chromatography-B, Journal of Chromatography Science, Journal of Pharmaceutical Analysis, Biomedical Chromatography, etc. So moving to the third question to Dr. Vikas Trivedi is that how can we decide for requirement of a chiral or chiral bioanalytical assay for new chemical entities? Over to you, Dr. Vikas Trivedi.
Thank you, Amal, for your nice and wonderful introduction. So the basic the requirement for the chiral and the achiral estimation in the bioanalytical assay, it's totally depend upon the molecule structure, like its stereochemistry and how many chiral center are available, and identification of the chiral center and the isomer of specific NC with reference to it route of synthesis. Where a racemate is used, then pharmacodynamic and pharmacokinetic study for both isomers in preclinical and then clinical state would be accepted or needs to be evaluated. So based on the overall evaluation of the preclinical and the clinical study data and the interpretation of the results, if the both isomers showing the different pharmacokinetic profile or difference in the pharmacodynamic results and having I can say having the difference in the exposure ratio like AUC of both the isomer by its rate of absorption. So in such cases, the analysis of NC should be performed using the chiral bioanalytical assay. If all this requirement does not match the acceptance, then we need to perform the analysis by using the chiral assay. Thank you, Emma.
Thank you, Dr. Vikas Trivedi, for answering the questions. Now, my fourth question is the presenter, Dr. Jairaj Chudasma. The question is that how to handle such condition where limited information of metabolite is available and analysis of clinical samples at high priority. Over to you, Jairaj.
Thank you, Aman, for asking a question. In few scenario where full characterization of metabolite is not available or it has some very limited information. So in that case, CRO has to establish the purity test using LC-UV tactics. After ensuring the purity, the metabolite can further use for the testing of clinical study samples. And apart from the potential interference of metabolite on the quantification of analyte needs to be checked, where exploratory estimation of metabolite is required in study sample analysis. Thank you, Emil.
Thank you, Jairaj, for answering the questions. Thank you all panelists for your active participation in the panel discussion and nicely answering the most of our audience questions. I would like to take a pause over here. We understand this session has kept us all well engaged throughout, but certain limitations of time, we would not be able to take any further questions at this point of time. But please be rest assured that we all be sending the answer to each and every questions of yours via emails. Also, we will be sending across the recorded link of this webinar on all the registered email IDs. Lastly, I, Hemal Kulani, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out time to be a part of this webinar. I would also like to extend our gratitude to Jairaj Chudasma for being such a wonderful presenter and giving us all great learning today. We at Veeda Clinical Research earnestly look forward to host you all again. Till then, take care and stay safe, everyone. Thank you.